I am Dr. Khokon Kumar. I have a Christian name, Dr. David Brun. I'm currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is Roles of TXNIP or TBP-2 of VDUP1 in heart attack and in the prevention of heart failure. Cardiovascular diseases are the leading causes of morbidity and mortality in the world today. Heart attack and subsequent heart failure accounts the major part of deaths from cardiovascular complications. Still today, it is difficult to predict when a person will commit heart attack. But recent breakthrough in medical sciences show great promises, while heart failure in humans may be completely preventable in near future following myocardial infarction or heart attack. It is to mention here that there is no treatment available today in the world to prevent heart failure with medications. Christine J. Reis et al. Published a research article in October 2019 in the Journal of Nature Communications entitled SR 9009 administered for 1 day after myocardial ischemia reperfusion prevents heart failure in mice by targeting the cardiac inflammation. The roles of inflammatory responses and recruited immune cells are considered as the critical factors for fibrosis, cardiac remodeling, expansion of infarct sizes, and heart failure after heart attack. Inflammations are now known as the central regulators of innate immune response, which work in a cooperative manner with the adaptive immune arm or antibody mediated immunity of the body. RAB is a transcription factor that negatively regulates the NLRP3 inflammation and the transcription of pro-inflammatory cytokine interleukin 1 beta, which are processed by activated inflammation into active interleukin 1 beta. The group used a pharmacological agonist SR 9009 for RAB to limit the degrees of inflammation, and recruitment of immune cells in the damaged site of the myocardium. They found that treatment for just 1 day with SR 9009 benefits the healing of the vulnerable infarct that arises after myocardial infarction or heart attack. To mention here that RAB is a circadian factor. From their landmark experiments, it is clear that the success in limiting the inflammation activation and inflammatory process determine the fate of the healing process of an infarct after a heart attack. But the limited participation of the immune cells are also required in the healing process. Now it would be nice to discuss briefly on the activation process of inflammation that lead the subsequent inflammatory process. Rangbin Zhou et al. Published an article in December 2009 in the Journal of Nature Immunology entitled Thioredoxin Interacting Protein Links Oxidative Stress to Inflammation Activation. They reported that TXNIP directly interact with NLRP3. Uric acid crystals induce the dissociation of TXNIP from thyroidoxin in a reactive oxygen species sensitive manner and allowed it to bind to NLRP3. TXNIP deficiency impaired the activation of NLRP3 inflammasome and subsequent secretion of interleukin 1 beta. In 2010, G2 Saxena et al. Published an article in the Journal of Biological Chemistry with the title Intracellular Shuttling and Mitochondrial Function of Thyroidoxin Interacting Protein. They found that TXNIP is primarily localized inside the nucleus of beta cells in the pancreas, and oxidative stress leads the shuttling of TXNIP to the mitochondria. TXNIP binds with thyroidoxin 2, TRX2 inside the mitochondria and reduces the ASK1 binding to TRX2. ASK1 gets phosphorylated then and initiates the death of insulin-producing beta cells in the pancreas with cytochrome C release and caspase 3 cleavage. TXNIP overexpression and TRX2 but not cytosolic TRX1. Silencing mimic these effects. This suggests that loss of insulin-producing beta cells in diabetic patients is caused by the mitochondrial TXNIP, mainly inhibiting the antioxidant mechanisms and increasing the oxidative stress. Troy Lane et al. Published an article in March 2013 in the Journal of Frontiers in Physiology with the title TXNIP Shuttling, Missing Link Between Oxidative Stress and Inflammasome Activation. They described that TXNIP shuttling into the mitochondria increases the ROS production and high accumulation of ROS inside the mitochondria. Increased ROS in turn oxidizes mitochondrial DNA also. Increased ROS activates the NLRP3 inflammasome complex and initiates the inflammation in the body. Oxidized mitochondrial DNA can also directly interact and activate the NLRP3 inflammasome complex. They also suggested that inhibition of translocation of TXNIP into the mitochondria may work as a great therapeutic approach to deal with the diseases where unexpected inflammations are involved. They also discussed that when there is ROS in high enough levels inside the mitochondria, TXNIP again translocates from the mitochondria to the cytoplasm and directly interact with the NLRP3 inflammasome complexes. In diabetes, inflammation is an important pathological causative for the loss of insulin-producing beta cells in the pancreas. So it can be demanded that translocation of TXNIP into the mitochondria also regulates the inflammation in diabetes and other diseases. Some recent advances can be mentioned here to signify the importance of TXNIP in heart attack and the prevention of heart failure. Yang Cheng et al. Published an article in October 2018 in the Journal of the American College of Cardiology entitled GW29 E1784 Cardiac Fibroblast Specific Deletion of Thyroidoxin Interacting Protein TXNIP exacerbates ventricular remodeling and dysfunction after myocardial infarction. Yuxian Tai et al. Published an article in February 2020 in the Journal of Molecular and Cellular Cardiology entitled M2 macrophage-derived exosomes carry microRNA 148A to elevate myocardial ischemia reperfusion injury by inhibiting TXNIP and TLR4 NF-kappa B NLRP3 inflammasome signaling pathway. E. Liu et al. published an article in July 2014 in the Journal of Basic Research in Cardiology entitled TXNIP mediates NLRP3 inflammasome activation in cardiac microvascular endothelial cells as a novel mechanism in myocardial ischemia reperfusion injury. Zhu H.J. et al. Published an article in December 2015 in the American Journal of Translational Research entitled Upregulation of microRNA 135A products against myocardial ischemia reperfusion injury by decreasing TXNIP expression in diabetic mice. Xiao Gao et al. Published an article in June 2019 in the Journal of Cardiovascular Research entitled TXNIP RAID1 signaling and excessive autophagy, a novel mechanism of myocardial ischemia reperfusion injury in mice. Jun Yoshioka et al. Published an article in December 2011 in the Journal of American Society for Clinical Investigation entitled Deletion of thyroidoxine interacting protein in mice impairs mitochondrial function but protects the myocardium from ischemia reperfusion injury. Ramin Rao et al. Published an article in May 2020 in the Journal of Thrombosis Research entitled Rivaroxaban ameliorates angiotensin 2 induced cardiac remodeling by attenuating TXNIP TRX2 interaction in Kai mice. Finally, I would like to wish a very happy, prosperous, and dignified life for myself and for you, everybody. Thank you so much.